Hello, good morning, everyone. This is Thomas from Safeter speaking. I'm the Director of Business Development at Safeter, and I'd like to welcome you to our webinar organized jointly with Swift Biosciences. The first part is going to be held by my colleague Drew McCusick from Swift Bio, who is going to give you a quick introduction to the company and to the product portfolio. And then in the second part, I would take over and I would present our company and our tools and services for interpretation of NGS data. The webinar is being recorded for your future reference. And also there is a section for questions. So feel free, to paste your questions in the Q&A section. And we will discuss your questions at the end of our presentation. Our presentation is going to take about half an hour, I believe. The first part is going to be held by Drew McCusick from Swift Biosciences. And so, Drew, please, over to you.
Okay, thank you, Thomas. Go ahead and share my screen here, or are we integrated in here? Okay, here we go. So thank you, Thomas. Yes, we're pleased to talk to you this morning and share a bit about the Swift portfolio first, and then the products which generate NGS data from Swift that feed well into the VarSome platform. So I'll just start by introducing this in terms of kind of how much of the genome or transcriptome that you may be interested in sequencing. So it's useful to kind of think about this in terms of this scale. On the far left here is whole genome sequencing, where, for the case of the human genome, for example, it's a 3 gigabase genome. And so there's lots of data to work with, and generally at lower depth due to sequencing costs. And then as we move to the right, exome and hide capture sequencing allows you to sequence quite a bit less of the genome. The exome is about 2% of the whole genome, but which informs you as to the genetic information for all the expressed genes. And then there's other hide capture panels that will function in that realm as well to help you target your sequencing data just in the regions of interest. And then on the far right is Amplicon sequencing, and this is where you are looking only at maybe only a few genes, a few kilobases of data, very targeted sequencing to drive your sequencing costs down when you know exactly what you're looking for, typically in more clinical scenarios. And so Swift has a variety of different library prep kits that will function to convert DNA or RNA into the NGS libraries that can produce data across this range of information. And so what we're going to be focusing in on just a little bit more detail today is these two on the right. The hide capture products and the Amplicon products from Swift, which are the kits that produce data that is supported through the analysis platform of the Safeter VarSome. So just a quick word on our hybridization capture panels. This is a process by which you are enriching out of your whole library just the regions of interest. And so here we're just showing you the kind of workflow on the left, how we do this capture with the probes, pull them out with a biotin streptavidin process, and do a post-hide capture PCR to do the sequencing. And so on the right here in this table, we're showing you the 3 different panels that Swift has available. Our most popular is the Swift exome. And that's where, as I mentioned, you're sequencing all the coding regions of the approximately 20,000 genes in the human genome. And then we have a more targeted panel for pan-cancer. This is just under 1 megabase, and it does whole coding sequence of 127 oncology-related genes. And then on the far right, we have an inherited disease panel, which about 11 megabases of space that's pulled down, and this is for mostly germline conditions that are inherited diseases. We do have some competitive information down here just showing that with this bait capture product from Swift, we have a higher percentage on target than several of these other competitors. So it's important to know that this is just the enrichment step of the workflow. It's what allows you to target, and it dictates what data you'll be working with in the VarSome platform. But also upstream of this, you have to first create an NGS library. And so I just wanted to spend 1 minute here talking about our most popular NGS library product, which is the Swift 2S Turbo. And so just to put these workflows together, you would first start with your DNA. The Swift 2S Turbo product is an enzymatic fragmentation, repair, and A-tailing step all in one, followed by a ligation step where you can ligate either truncated adapters from Swift or your own. It's flexible. There's a version called 2S Flexible on the right here where you can ligate your own full-length adapters. And then in the final step, you can perform a PCR. In the case of the downstream hybridization capture for those exome, pan-cancer, and inherited panels that I just showed you, you'll absolutely have to do some PCR because you're just capturing a very small percentage of the sample with those probes. And so you need to produce enough library to do that downstream capture and have enough material. And so this is, as I mentioned, a very popular product that we have for both whole genome and exome and hype capture sequencing. It's a very low cost, very quick prep. And so this is just kind of, these are just example kind of US list prices on the left here. But what really makes this product special is that it's much faster and lower cost than its competitors. And then another thing that's important, that becomes very important in the library prep stage, before we get to the data analysis through VarSome, is that in these preps, if you're not capturing the full complexity of the sample, then with the library prep, then when you go to do the hybridization capture to enrich just the sequences of interest, you're going to be putting at risk your ability to actually capture that complexity. And so on the left here is a competitor that also uses an enzymatic fragmentation step at the beginning. And you can see that for the lower GC content on the left-hand side of the graph, so this is AT-rich parts of the genome, there's this huge dropout that you can see illustrated with the blue circles, which is the normalized coverage relative to how well represented that part of the genome should be. So you're actually looking for kind of this straight line across one here. And so you can see that a competitor's product is still doing this enzymatic fragmentation-based library preparation, but at the expense of this coverage of the lower GC content. And so then you can imagine these molecules are not available, or they'll have a lot of bias in terms of their availability for the downstream capture and sequencing depth, which affects ultimately your ability to call variants or do analysis in those regions. In contrast, on the right here, the 2S Turbo product still produces a very even coverage across the entire range of ATGC base composition content in the genome. Okay, so switching gears here, this is now, if you remember at the start of the presentation, the Amplicon panels from Swift, which allow you to interrogate up to a couple hundred kilobases of content in the genome. And so Swift has a simple single tube 2-hour workflow, which is very convenient, very fast for interrogating those sequences that you're interested in. This functions through a PCR step. And so anytime you're doing a multiplex PCR where you'd like to use more than a handful of primer pairs at a time in the same reaction, you have to have a special technology in place to prevent the unwanted formation of primer dimers. And so Swift has a patented multiplex PCR step And then this produces all the different amplicons of interest. There's a cleanup step, and then there's a ligation indexing step where we ligate the adapters for Illumina sequencing. And then after that, there's a final cleanup, library quantification, sequencing, and then this is the FASTQ data from the sequencer can be uploaded directly into the Safeter VarSome platform. These products, we have a number of both kind of core or catalog panels, as well as the ability to do custom panels. As they are here, the limit of detection for mutations is a 1% variant allele frequency. And so this is to say if you have 100 copies of the genome and just one sort of needle in the haystack copy of a gene, a mutation-bearing copy, that will be detectable in that background. These panels, we do a high degree of quality control and balancing to confirm that they are performing at greater than 90% on target and greater than 90% coverage uniformity. And so these are metrics that confirm that you're amplifying the regions of the intended genome that you wish to only, and also that across the different regions that you are amplifying, for example, these amplicons, dark blue, light blue, and purple, that they're relatively evenly represented within each sample. You don't want to have one particularly dominating or dropping out. And so we have also in this workflow the ability to tile amplicons across exons. And so this can be really important because unlike some of the competing amplicon-based library preps, you know, we allow you to put all of these primer pairs. In this example, there's a gene ESR1 that's relevant to breast cancer. And, you know, in genes like this or TP53, where a loss of function mutation across anywhere in the gene might be important to the function or loss of function of that gene, it's important to cover the whole thing. And then if you're working with a limited material like cell-free DNA, where you maybe only have 10 nanograms, it's ideal if you can interrogate that whole exon in a single PCR reaction. And so the Swift Excel Amplicon panels allow you to do that. You can see here we have, for example, 5 different primer pairs tiled across this exon in ESR1, whereas competing library preps will have you split these into 2 different PCR reactions. And so this is just a brief comparison again of the different library preps for Amplicon that are predominant in the market, and how Swift's on top here is shorter and with fewer tubes. This is a snapshot here just of the different catalog panels that Swift has available, and each of these is supported by the VarSome platform for data analysis. So our most popular is the 56G panel, which interrogates 56 oncology-related genes, including full exon coverage of TP53. And then we also have EGFR pathway, BRCA panels, and then TP53 and the cystic fibrosis gene in a panel. And then on the right side, we have some groupings of genes based on the disease relevance that are also available. If you do wish to do a custom panel with us, there's a process by which you submit those targets to us. Swift runs our design software to produce a design report. And once we agree on the design consensus, we'll go ahead and synthesize the primer and optimize the panel performance. There's a wet lab testing process that we do to optimize the panel against control DNA. And then we'll provide that assay to you so you can implement it. Custom panels are also supported by the VarSome platform. This is just a quick snapshot of the 56G panel. This is also available in the data sheet for this product on our website. We're just showing you which genes are there. You can look at the amplicon size and average of 138 base pairs, which confirms that it will be compatible with liquid biopsy samples like cell-free DNA, as well as degraded tumor samples like formalin-fixed paraffin-embedded samples. And in fact, just to share with you a quick publication highlight, Strand Life Sciences in India is one of those who has published using the Swift panel and looked at that exact function of the assay, because the 56G panel can function with both cell-free DNA as well as FFPE material. they were looking at the concordance between those two sample types and made this publication. Similarly, MD Anderson here in the United States was using a custom panel and looking at plasma tumor concordance as well. So these assays are very well suited for those types of studies. There's a host of other publications I encourage you to look at on our website. And so here's just a few kind of snips of those headlines, those article publication titles. And so just to conclude here now, I'm going to hand the podium back to Tomas, but I just wanted to mention that Swift manufactures and sells our library prep kits, and we're also very pleased that VarSome has allowed us to partner with them to act as a resale distributor of their tokens. And so if you're working with Swift directly in the US or one of our distributors abroad, we have a catalog number set up, ALVS48, which allows you to trial and purchase these tokens directly from Swift as well. And so the first token we offer at an evaluation discount. And so if you're interested in that, please inquire. And then as well, if you're looking to bundle the tokens with our library prep kits or to purchase a quantity of tokens and kits over time, then we're happy to work with you there. So I think that wraps up my presentation for today. So thank you, and I'll be here till the end of the presentation. So if there's any questions about the Swift library preps, please feel free to ask, and otherwise I'll hand it back to you, Tomas. Thank you.
All right, Drew. Thank you very much for a comprehensive, nice introduction to Swift Biosciences and to the portfolio of your gene panels. Now I'd like to take over, and I'd like to give you a quick introduction, to our company as well as to our products and services. And then in the second part of my presentation, I'd like to perform a brief demonstration of capabilities of VarSome Clinical. So we, Safeter, we are a precision medicine company and bioinformatics company based headquartered in Switzerland, in Lausanne. We are on the market since 2014, and we develop tools for processing and interpretation of NGS data specifically for clinical purposes, although we also serve research customers. Obviously, there are many challenges in bringing NGS technology to the clinical practice. However, from our perspective as a bioinformatics company, we are focusing on 3 challenges in particular. The challenge number 1 is the fact that the annotation data are fragmented and spread over many data resources. There are possibly hundreds or tens or even hundreds of data resources where you can go after a specific piece of annotation data. However, the interpretation process can be seen as a puzzle game, right? I mean, to get a complete picture, you need to place all the little pieces together. And so that's really challenging. Challenge number 2, it's a never-ending process. I mean, our understanding of the mechanism of diseases is becoming better pretty much every day. And so what was true or what was valid yesterday or today, doesn't have to be valid tomorrow or the day after. And challenge number 3, there is a very little standardization in terms of interpretation of pathogenicity, meaning there might be 2 laboratories working and interpreting the same variant very differently as a consequence of having access to a different annotation data. So we are focusing, on these 3 challenges. And so these 3 challenges are the reason why we have created VarSome in the first place. And so VarSome, I guess some of you might have seen it before. VarSome, it's free and it's open platform. It's a global community and genomics knowledge base. And so what VarSome does in the first place is that it aggregates and cross-references publicly available annotation data. So Currently, you can find on VarSome annotation data from more than 50 public data resources. Data resources such as, ClinVar, GNOME AD, all the transcripts from RefSeq ensemble, annotation data for copy number variation, structural variation, expression profiles, annotation databases for cancer, such as ICGC, PMKB, CIVIC, list of publications, a list of drugs, list of clinical trials, in silico predictions, conservation scores, clinical data resources for genes, phenotypes, and diseases, and so on and so on. Over 50 data resources matched together. On top of that, we keep the annotation data on VarSome always up to date. So whenever there is an update in ClinVar, let's say, or GNOME AD, we get the notification, we get the update, we process it and make it available on VarSome in a very short turnaround times. So you have always the latest annotation data from all these 50 data resources. And also we are implementing guidelines for standardization of interpretation of pathogenicity. So currently you can find on VarSome the automated interpretation of pathogenicity based on well-known ACMG guidelines. I will talk about it a bit later. We are also now implementing guidelines for somatic interpretation, such as AMP and CCN or ASCO. However, VarSome, the free platform, it's not only about data integration and keeping the data up to date. It's also about you, about our global community. So currently we have on VarSome over 200,000 users using the platform pretty much every day. And as a consequence of it, there is also a growing number of contributions flowing in from our global community. So on VarSome, you can find contributions in terms of publication links or links for functional studies, classifications. VarSome users also start new collaborations on VarSome. So there is there is additional layer of data which is very unique to VarSome and which comes from our global community, which further enriches the knowledge base. On top of the free platform, we have developed a VarSome Clinical. So VarSome Clinical, it's already a professional service, allowing you, to interpret your NGS data, starting either from FASTQ or VCF. And at the end of the process, you get a clinical report. So VarSome Clinical is certified as an in vitro diagnostics medical device according to the requirements for CE-IVD devices. So it can be used indeed in clinical settings. Our platform is also HIPAA compliant, which is very, very relevant for our US-based customers. Our company is, of course, also GDPR compliant, and we are certified with ISO certificates for data quality and data security. With VarSome Clinical, you can process your NGS data in 4 steps. Step number 1, data upload to our servers. You can either start from FASTQ or VCF, and you can process any kind of NGS data, be it a gene panel, like a commercial gene panel, such as Swift Biosciences, or custom built a gene assay, built based on the Swift Biosciences technology, or it can be exome or genome, or any kind, simply said, any kind of NGS data. VarSome Clinical offers a wide range of pipelines covering pretty much all the clinical use cases. So we have pipelines for germline samples, for somatic samples. We have a pipeline for example, de novo variants in trios, for carrier risk screening for couples, a pipeline for tumor normal pairs, including a pipeline for a copy number variation, structural variation. And the latest addition to the portfolio of pipelines is a pipeline, supporting UMIS, a unique molecular identifiers, allowing the detection of low frequency variants typically found in somatic samples. VarSome Clinical comes with a website interface, which means you don't need to install anything locally. Everything runs on our server, and you access the platform over the internet as if it was a regular website. So you don't need to worry about the underlying hardware infrastructure. You don't need to worry about updates, about security issues. We take care of all these things. And in the last step, you can generate a clinical report which can be fully customized in terms of the look and feel of it. So you have complete control over the design of it, as well as you also have control over the data to be included in the clinical report. So you can decide, for example, whether you want to include clinical trials or not, depending on your specific workflow. You know, the challenge here actually is the interpretation, right? I mean, to do the primary and secondary, you know, part of the pipeline, to do the read alignment and variant calling, that's relatively easy. However, when it becomes to be really challenging is the interpretation part of the pipeline, and I believe you would agree. And that's what we are specifically focusing on. We are focusing on the interpretation part of the pipeline. VarSome Clinical, it's not a complete replacement for a bioinformatician. It's actually a productivity tool allowing your bioinformatics team to be more productive, more systematic, and spend, you know, the energy where it is really needed. So here I just wanted to share with you, you know, our list of clients here. So as a Swiss company, we have a number of customers in pretty much all the European countries, including Middle East. And we have quite a few customers also on the global scale. We have a number of customers in the US, in Latin America, India, Southeast Asia, Hong Kong, Singapore, and so on. From the business model perspective, there is no license for VarSome Clinical. VarSome Clinical charges on a per sample basis, depending on the type and size of your sample. So when it comes to the FASTQ samples or FASTQ files, the price depends on the number of megabases in reads. And when it comes to the VCF files, the price depends on the number of variants. So there is no license fee, only sample fee, and all the pipelines are included. There are basically two ways how you can use VarSome Clinical. You can use it either as the so-called standalone solution. So with the standalone solution, you can process any kind of NGS data. In other words, the platform is open for any kind of NGS data, be it a gene panel, exome, or genome. And basically, you know, we or our distributor would send you an invoice on a monthly basis, depending on the usage of the platform. So this is nothing else than the regular, you know, pay as you go model. However, what could be even more interesting, I guess, you know, for most of you, is the bundled solution. So the bundled solution, it's a form of prepayment, and Drew mentioned that already in his presentation. So this is where Swift Biosciences, you know, comes into the picture. We have become partner with Swift Biosciences, and what we do is that we bundle our products together. Swift assay, be it Amplicon based or capture based, bundled with VarSome Clinical for data interpretation and reporting. And this way we can deliver a truly complete solution, right? Starting with the library prep and ending up with the generation of clinical report. So this is a form of prepayment where you can prepay, you know, certain number of samples belonging to a specific assays, and we have number of partners. Of course, with biosciences, it's the most important partner currently for us. And also for your information, VarSome Clinical can be also installed on your premises, on your own server, which makes sense only for a large scale sequencing or diagnostics center. So this is my quick introduction to our products and or to our company and to our products. And now in the second part of our webinar, I'd like to show you our platforms in real time. So first, I'd like to show you the free platform, VarSome. And so, for example, here you can see the results for a particular variant in a BRAF gene. Yeah, so I'd like to guide you now through the result page very quickly. So first, here you can see the common information for the variant, chromosomal position, reference, alternate sequence. Here you can see the ACMG-based interpretation of pathogenicity. I'll get back to it in a while. Further, here you can see the gene landscape, for example, where you can see all the variants within the given gene, with all the supporting, pieces of evidence coming from GNOME ID, UniProt, ClinVar, DBSNP, ICGC, and so on. Here you can see the contributions from our global community. So in this case, there are 38 contributions in terms of, publication links, classifications, or discussions. Going far down the page here, you can see the list of publications, structural variant browser with all the transcripts, with all the structural variants coming from ClinVar, exact CNVs, DGV, all the transcripts from RefSeq, Ensemble, ClinVar entries. So this is a particularly well-studied variant in a BRAF gene. So there are quite a few entries in ClinVar, UniProt variants, population frequencies broken down by ethnicity and gender. PMKB is quite important, annotation data resource for somatic, samples. PMKB stands for the Precision Medicine Knowledge Base, which is a manually curated list of variants by Weill Cornell Institute in New York, which also includes, list of drugs as well as list of clinical trials. And then number of other somatic data resources such as ACGC, Cosmic, Civic, clinical data resources such as Genomics Data Commons, and at the end of the page, number of pathogenicity scores and conservation scores. So indeed, as you can see, on a single page, you get wealth of annotation data coming from more than 50 data resources plus contributions from our global community. So it's a very robust knowledge base, which we leverage for the automated interpretation of pathogenicity based on ACMG guidelines. So here I'd like to stop for a while and I'd like to tell you a little bit more about it. So in this case, you can see that 6 ACMG criteria fired automatically based on the aggregated evidence coming from all these public data resources. So in other words, VarSome's implementation of ACMG guidelines, it's very robust, right? It leverages, wealth of public data plus, contributions from our global community. Second, it's also very transparent implementation, right? Because for each criterion here, it tells you in a great detail, in plain English, why given criterion fired or didn't fire. So this is something that allows you to review each particular situation and make possibly your own judgment call in line with your best knowledge. And it's also very flexible implementation of ACMG guidelines because it allows you, to specify the transcript, for example, which may affect the final verdict, Or you can also specify, the weight of the evidence as a very strong moderate or supporting, again in line with ACMG guidelines. So in the case of PP5 criterion, for example, here, that we always, for any criterion, we consider a multiple pieces of evidence wherever possible. So in the case of PP5 criterion, for example, you can see that it considers ClinVar entries, UniProt entries, as well as contributions from our global community. And so 3 pieces of evidence are considered for evaluation of PP5 criterion, and the same procedure applies, for all the criteria. Not all the criteria can be fully automated. Certain criteria require patient-specific information, patient-specific information, which is not obviously available to us. And so it's in those cases, it's you who is supposed to make, the judgment call in the case of BP2, BP5, PS4, PM3, PP4. So this is the story, behind the free platform, a knowledge base, community, and community around it. Now, on top of it, we have built, the clinical platform called VarSome Clinical. And so with VarSome Clinical, you can process any kind of NGS data, starting either from FastQ or VCF. So now I'd like to show you, the core features as quickly as possible. A full demonstration would take me about half an hour. It's quite complex platform, but I'm going, to focus on the most important aspects of it. So let's say we are starting from FastQ. So here we shall specify the pipeline details, right? We shall specify whether it's a single sample, family trio, or couple for carrier risk screening, or whether it's a germline or somatic pipeline. Here you shall specify the assay. So here you can see a number, not a number, all the Swift Biosciences assays, including 56G gene panel. You can process any kind of NGS data, be it like either based on Amplicon based technology or capture based technology. When it comes to the custom gene panels, we need the BET file first. Here you should also specify the ethnicity of your patient, reference genome, and a couple of other things. Then on the left side of the screen, you shall specify the sequencing data, the FastQ files or VCF files, which you have uploaded previously. You can also provide sample IDs and descriptions, and you can also optionally type in the phenotypes and diseases of your patient according to the HPO, to the human phenotype ontology, such as, I don't know, retinopathy and so on. And so later in the process, you can use these terms for variant filtering and variant prioritization. So once you set up all these details, you can proceed and you can start the analysis. The analysis takes about 10, 15 minutes, depending on the size of the gene panel. When it comes to exome, an exome analysis takes about half an hour, and analysis of a whole genome takes about 4-5 hours. So as you can see here, you can see an example of a Swift 56G panel analyzed with VarSome Clinical. I'll get back to it a bit later. First, I'd like to show you actually a whole genome sample, because that allows me, to show you all the core features of the platform. So here you can see this is a whole genome sample with almost 5 million variants. So this is quite typical healthy genome. The variants are sorted by pathogenicity according to the ACMG guidelines. Again, we follow here, the same interpretation procedures as on the free platform. Further, here you can see, for example, the list of ACMG criteria which fired for each individual single variant out of these 5 million. AGVS term, transcript, exon number, gene, inheritance, function. In terms of function, VarSome Clinical annotates against all the transcripts available. So whether it's a splicing, non-coding, coding, missense variant, and so on. Zygosity, frequency, this is the population frequency according to the ethnicity. Allelic balance, this is a percentage of reads supporting the given variant. And coverage. Coverage, this is a link which takes you, to the JBrowse view where you can see the graphical representation of reads. There is also IGV available as an option or as an alternative to JBrowse. Now, around the variant table, you can see tabs with annotation data coming again from all these 50 or more public data resources. So for example, here you can see the frequencies, allele frequencies, clinical data, clinically relevant data for genes, phenotypes, and diseases, list of transcripts, genes, HPO terms again. I'll get back to those a bit later. Here you can see the ACMG-based interpretation of pathogenicity, the region browser, where you can see again all the variants within the given gene, ClinVar entries, caviar entries, population frequencies, Bravo population frequencies, somatic data resources, ICGC, GDC, in silico predictions, conservation scores, publications, and also contributions from our global community. So again, a wealth of annotation data available across these tabs. Now, for example, let's take this variant as an example. This is a pathogenic variant, right, according to the ACMG guidelines. However, if you don't agree with the default verdict, you may reclassify the variant as a benign or variant of uncertain significance. Or you can set up a fully customized classification, such as variant important from a pharmacogenomics perspective or variant which needs to be discussed a bit later. And then again, you can apply these custom classifications to your variants here. So this is the way how you can build over time your private database of classifications. These custom classifications stay with the variant forever, basically, unless you change it or remove it, which also means that whenever you upload a new sample with the same variant, you will see here the same classification mark along with the name of the person who set up the classification. What we also do is, so here under samples here, you can see the list of samples sharing the same variant in a either homozygous or heterozygous form, right? So what we do actually is that we link all your samples, together on the variant level. So this is something what allows you, to go back and forth between your past cases and current cases, compare the patient phenotypes and diseases, and ultimately increase the diagnostic yield. So VarSome Clinical, it's all about increasing the diagnostic yield. It's all about productivity and being, organized and focused on what's really important here. So with these, custom classifications and sample cross-referencing, that's how we call it. These two features taken together allow you, over time to increase, the diagnostic yield for your patients. We can even take your manually curated list of variants you may have in an Excel sheet, for example, and we can import it privately, to your account with VarSome Clinical for the purposes of variant classifications. Here, under the Analysis Actions menu, here you can see a number of QC reports. So, for example, here you can see the basic QC report, which summarizes, the sample details, details of data resources used for interpretation, summarization, of the alignment step, summarization of the primary and secondary part of the pipeline, summarization according to the coverage or sequencing depth, summarization according to the function, summarization according to each individual ACMG criterion, and at the end of the page, summarization in terms of function. So this is the basic QC report. However, there is a number of other reports. So for example, you can also download the BAM file, index BAM file. You can download the VCF file with full annotations, or you can also access here the coding coverage report. So the coding coverage report, it's a very comprehensive report where you can see it's an Excel sheet, obviously. And so what you can see here, you know, you can find here, you know, the regions with a low coverage, or regions, you know, which have been skipped altogether from some reason. So this is the place. This is the place which allows you, you know, to troubleshoot issues either with sequencing or with assay design. And then there's a number of other options. I encourage you, you know, to check out these options a bit later. Now, filters. So we have 5 million variants, right? So the filtering is really important here. So here on the left side, you can access the filters. So you can filter your variants based on various criteria, such as ACMG rules, based on population frequency, based on ClinVar classification, chromosomal position, pathogenicity, zygosity, function, whether it's frame shift, nonsense, 5UTR, splicing variant. You can apply a gene list as a filter, call status. You can filter your variants based on coverage or allelic balance, and number of other filters here. So just to give you a quick example how it works. So here, let's say we are interested only in rare variants with a frequency less than 1%. So now we are setting up a filter. A filter set may consist of several filters, such as frequency filter, and the other one can be a filter for pathogenic and likely pathogenic variants. So now we have 2 filters within a filter set, and now we can apply it to our sample here. So again, as you can see, everything is happening really fast, right? Don't forget that now we are working with a whole genome sample with 5 million variants, and still everything works super fast. And so here you can see the results of the filtering based on the frequency filter. You can see that over 4.5 million variants are gone. And based on the pathogenicity filter, additional 339,000 variants have been eliminated from the list, and we are left with only 5 variants in the variant table. You can always, you know, deactivate certain filters or reactivate them again. And the results in the variant table here update dynamically. In fact, this is the reason why we call these filters dynamic filters, because you can easily modify parameters and thresholds, you know, and the variant list updates dynamically. You can even go one step further, and you can modify the filter set. You can now include additional filter based on ACMG criterion PP5 with a supporting strength of evidence. So now we have 3 filters within the filter set. Again, we are going to apply it, and now you can see that we are left with only 2 variants in the variant table. So these are the dynamic filters. However, that's not all. VarSome Clinical offers even more comprehensive filters called algorithmic filters. So algorithmic filters are super powerful filters which can be fully customized according to your specific workflow. So here you can see several common examples of algorithmic filters, just for your idea, for your you know, just for your information about what's available here. So for example, with algorithmic filters, you can do de novo variants, call de novo variants in trios, or you can perform a compound heterozygous kind of analysis, or you can perform segregation kind of analysis, and so on. So these filters can be fully customized specifically, you know, to your needs. And then you can combine them. So here, for example, here you can see an exome trio with 290,000 variants, and then within the original analysis, you can see a number of sub-analyses. So each sub-analyze corresponds to the results of an algorithmic filter. So for example, here we can see de novo candidates de novo candidates in exome trio. There are 260 variants, and then of course you can combine, you can lay over, you know, the dynamic filters to narrow down, you know, the search space as much as possible. So now the last missing piece is the reporting. So here I'd like to show you just very quickly how the reporting works. We are running out of time, unfortunately. As I was saying, this is a complex platform. So once at the end, you know, I mean, the process is fully automated until you get, you know, the list of variants with full annotations. Now, when it comes to reporting, it's your responsibility to decide which variants you are going to report or not. So once you take the decision to report it, you select the variant for export, and then you can proceed and you can generate a clinical report. So as I was saying before, this is kind of like an interactive reporting where you have a complete control over various aspects. So here, for example, you can upload your own logo. You can update, you know, the header. You can set up your own fonts, color, and sizes in line with your branding policy. So basically, what you need to do initially is to set up a template, which you can use later, you know, for all your analysis. And then here on the left side here, you can see widgets with annotation data, with pieces of annotation data, and it functions as a drag and drop editor. So for example, here you can drag in, you know, the content area, and then you can drag in, you know, the information about the variant, clinical information about the gene, clinical. Then we can also include the list of drugs, list of clinical trials. Oh yeah, additional clinical data resources for genes, and then we can also include the list of publications. So everything is fully editable. You know, the report can be fully translated. I mean, the headlines and titles can be fully translated. The content itself, it's always in English, of course. So here you can provide your custom comment for the report. And then here you can see the information for the variant, AGVS term, zygosity, exon number. Here you can see, you know, the list of phenotypes and diseases, mode of inheritance, list of drugs, a list of clinical trials, and at the end, list of publications. Everything is fully editable. So in case you don't like this publication, you can remove it, and you can include additional publication according to your preferences. So you have complete control over the report. You just need to set up a template initially, and then you can use it all over again. And then once you are happy with the report, you can download this PDF file or as a doc file. So here you can see a couple of examples. So for example, here you can see a report for the Swift Biosciences 56G panel. Please note, this is a default template without any styling. So here you can see the same thing as before, clinical information for genes, phenotypes, and diseases, list of variants here. Here's another report. Yeah. You can basically control, you know, which columns you want to include. So that was just a very quick introduction, you know, to VarSome Clinical. VarSome Clinical, it's a quite complex platform, allowing you, you know, to generate a clinical report in 4 steps for any kind of NGS data, be it a custom panel, commercial gene panel such as Swift Biosciences, exome, genome. It doesn't really matter. VarSome Clinical is clinically certified as a CE-IVD device. VarSome Clinical charges on a per sample basis. depending on the size of the sample, either in terms of megabases or number of variants. And you can also get it as the bundled solution in a collaboration with the Swift Biosciences, where you can basically prepay a certain number of analyses. This is what we call a token. Yeah, so you can basically inquiry for a token, allowing you, to analyze a given number of samples. So that's about it from my side for the moment being. I've tried really to focus, on the most important aspects. Otherwise, I could talk, for hours probably together with Drew. So, Drew, do we have some questions on Zoom?
I'm not seeing any here on my end. Says no open questions. But yeah, we appreciate the opportunity to share this with you guys. If you have anything to ask, we're here for another few minutes. I think one thing we could speak to as well, just briefly, is one of our customers who's adopted the joint workflow of the Swift Amplicons and the VarSome platform is doing so in an environment where they're very aware of the exact type of report they need to generate in order to have a certified test and the various reporting and kind of reimbursement structure they need to look for. And so that's one place where we've seen this partnership work really well is, they did a custom panel, as well as I believe they were working with our colorectal cancer panel and our lung cancer panel. It's a customer in Czech Republic. And in this case, the kind of adoption process looked like, trying the kit from Swift, making sure they could generate libraries first, making sure they could generate yields reproducibly. With some of these clinical samples, it's very difficult based on the material you're working with sometimes. And so we have a number of things in our kits that can help you qualify the input material accurately. And then after we sorted that out with them, they were making libraries very reproducibly and generating the data they needed to. And so that got them to the FASTQ stage, at which point they started using these tokens from VarSome for the primer trimming, alignment, variant calling, interpretation, and report generation. And I think what you mentioned, Tomas, about those earlier stages being relatively straightforward, I think is very true. As long as with the Swift Amplicon workflow, for example, as long as there's they're able to work through the protocol accurately. And sometimes, there's only 2 steps in the Amplicon kit, but sometimes there can be a small mistake made. And we have a technical support team that can get you through that stage if you have any issues. And then, like you said, the generation of the sequencing data and even the variant calling steps are relatively straightforward. But what's really powerful here is this ability to leverage all these different databases and annotations through VarSome Clinical in order to get something that's actually very useful from the sequencing data that you can act upon. So that's kind of how I would conclude. And again, thank you guys for joining. I don't know if you have any other closing remarks, Tomas. I don't see any questions still from the attendees.
No, it looks like we have covered, everything. There are no questions at all. So that's very rare, but it's a good sign, probably. Or I don't know. So no, not really. Thank you very much again, Drew, for joining our presentation today. I think it was quite nice and productive presentation. Also, thank you the audience for being here. And as I said, our webinar has been recorded, so I'll be in touch very soon with a follow-up email sending you the link for it, as well as additional learning resources. So thank you very much, everyone, and have a good day. Bye-bye.
Thank you. Bye-bye.